All right, so we're on chapter 10, section 3, and we're going to talk about all of the different enzymatic reactions inside of the citrate cycle. So the citrate cycle is 8 different reactions, but the really important ones are the 4 coupled redox reactions. So 3 here, 4, 5, oh, not 5, 6, and 8. These are the ones that are serving to transfer our pairs of electrons from our organic substrates, right, our reactants, to those energy carriers, NAD plus and FAD. And those are our oxidants, right? We also do make GTP in reaction 5, and that GTP is converted to an ATP. But those are our energy conversion reactions within the pathway. So remember that you had pyruvate, you converted pyruvate to acetyl-CoA. You got 1 pair of electrons transferred to NAD+, right? So when we talk about the citrate cycle, usually we include what happens in order to go from pyruvate to the end of the citrate cycle. So for the citrate cycle, what you're really going to end up getting, I'll put it over here, are 4 NADHs. You're going to get 1 FADH2, and then you get 1 GTP. And then you end up converting this whole thing. You go from pyruvate to CO2. Okay, that's where your carbons are. Your carbons are in pyruvate. Where do your carbons end up? Your carbons end up in carbon dioxide. So in order for a citrate cycle to go, it's aerobic. You need O2, right? And why do you need O2? Well, think about, remember we said O2 is our endpoint in our electron transfer, right? And so we really need a continual oxidation of NADH and FADH2. We have to keep passing those electrons along because we have to keep regenerating NAD plus and FAD. So if you don't regenerate them, if you don't drop them off, you're not going to be able to go back and do any more. So we have a cyclic pathway, and we'll talk about why that's really important. And there are 8 different reactions in there. Just like in glycolysis, right? We have 3 highly favorable reactions. And so if you look, if I could circle, they're in blue here. These are our highly favorable reactions, right? And so these reactions, you'll notice that they're only given on standard conditions. They're not actual conditions. And that's because it's really, really hard to assay inside of the intermitochondrial matrix without getting contamination from the cytosol. And so we don't really have real values for that. But what we think is that these reactions probably have delta G values, these 3 that I've circled, have delta G values that are, you know, significantly far away from 0, significantly less than 0. So they're negative. So they're very spontaneous. So these are our key regulators of the citrate cycle flux, right? They're all really close to 0. All of these reactions are really close to 0, except, right, the ones that we've circled, and look at this one, our one here at the end, this is a positive 29.3. Wow, how does that go? Well, the substrate, not the substrate, the product in this one, oxaloacetate, this substrate concentration is kept very low. And that's how this one proceeds. All the others will proceed, either they're really close to 0 and they're at equilibrium, or they have a very high negative delta G value, right? So when we think about this, right, you have to think in terms of oxidation reduction, right? And so do you have enough oxygen or not? In this entire pathway, if O2 is limiting, right, then we're going to start accumulating NADH, FADH2, and then when those things accumulate, we're going to have a feedback inhibition that's going to occur. So our O2 has to act as our terminal electron acceptor to replenish NAD plus and FAD. That's really, really important in order to make this cycle go. And so here's our whole cycle. And we're going to talk about this step by step. But it's good to have kind of a picture to know where you're starting from, where you're going to end up with. And I'm not going to talk in detail about this until the end. Okay, so what I want to show you is a quick little animation. All right, so here we go.
Citrate cycle. The citrate cycle is the metabolic engine of the cell. When energy in the cell is low, the citrate cycle is critical to the production of energy from fatty acids, proteins, and carbohydrates. The fuel for the citrate cycle is acetyl-CoA. The exhaust is carbon dioxide, and the work performed is the transfer of electrons. In each turn of the citrate cycle, one acetyl-CoA is oxidized, and 8 electrons are transferred to 3 NADH and 1 FADH2. In the process, 1 GTP is also produced by direct phosphorylation of GDP. The NADH and FADH2 are used to power the electron transport system, resulting in the production of approximately 10 ATP for each acetyl-CoA that enters the citrate cycle. The flow of acetyl-CoA into the citrate cycle is controlled by the pyruvate dehydrogenase enzyme complex. The pyruvate dehydrogenase complex has a molecular mass of almost 8,000 kilodaltons and contains nearly 80 individual protein subunits. Pyruvate dehydrogenase uses pyruvate from the glycolysis reactions and metabolizes it to acetyl-CoA, fueling the citrate cycle. The pyruvate dehydrogenase complex consists of 3 interdependent enzyme subunits that use pyruvate, CoA, and NAD+ as substrates, and in a 5-step reaction mechanism, generates carbon dioxide, acetyl-CoA, and NADH. 5 different coenzymes are needed to enable the enzymatic activity of the pyruvate dehydrogenase complex. Nicotinamide adenine dinucleotide, derived from vitamin B3, flavin adenine dinucleotide, derived from vitamin B2, coenzyme A, derived from vitamin B5, thiamine pyrophosphate, derived from vitamin B1, and lipoamide. The product of the pyruvate dehydrogenase reaction, acetyl-CoA, is used to fuel the citrate cycle. The first reaction in the citrate cycle commits to the acetate unit of acetyl-CoA to oxidative decarboxylation by catalyzing its addition to oxaloacetate, forming citrate. Oxaloacetate is regenerated in the last step of the cycle, so it can be reused as the substrate for the first step. Note that the metabolic cycle is referred to here as the citrate cycle, because citrate exists predominantly in the deprotonated form at physiological pH and is therefore not an acid in living cells. Other names for the citrate cycle are the tricarboxylic acid cycle, or simply TCA cycle, and the Krebs cycle, named after the biochemist who first described it, Hans Krebs. The citrate cycle contains 4 coupled redox reactions. They use the oxidized forms of the coenzymes NAD+ and FAD as electron carriers, reducing them to NADH and FADH2. NADH and FADH2 are then oxidized by the electron transport system, producing ATP. Citric acid is used commercially for a wide variety of purposes. It can be isolated in great quantities from the fungal mold Aspergillus niger using fermentation methods that exploit the citrate cycle reactions. High levels of citrate synthase are found naturally in the fungus. When the other citrate cycle reactions are inhibited under certain culture conditions, citrate is overproduced by the fungus and secreted into the culture medium.
Okay. Okay, so now we have kind of this nice overview of the citrate cycle. So let's go into it in detail. This first reaction 1 with citrate synthase, is super important. This one we said had a really low delta G value, right? So reaction 1, right? This very first one is irreversible. That's what that very large negative delta G tells you, that it is irreversible. And the point of it being irreversible is that we're really going to commit the acetate unit of acetyl-CoA. So this is the acetate unit of acetyl-CoA. We're going to commit this to oxidative decarboxylation. We're going to commit it to this cycle to make sure that we really go through the cycle. You want that first step to be irreversible so that we get the wheel rolling, basically. So it's irreversible because it's highly favorable, right? And then our other substrate here, oxaloacetate, is not only the substrate for our first reaction, but it is going to be the product of our last reaction so that it is regenerated throughout the whole cycle. And so one thing that's really kind of important to notice is that you see the carbons that are from the acetate unit are highlighted as to where they're added in the molecule. And so we're kind of going to track them as we go. And what we're going to notice is that the carbons that come in from the first cycle, from this cycle, are going to stay in the molecule. They stay in the molecule. You're even going to see them in oxaloacetate. And then when they go into the second round, then those carbons are going to turn into carbon dioxide in the next round. So you'll see that it takes a complete turn, and then you have to enter the second cycle in order to lose those carbons. So the ones that are in black here are the carbons that we're actually going to lose to carbon dioxide in this cycle. Okay, all right, so our second reaction, our second is reversible, a-Ketoglutarate. This is really a two-step mechanism. And it's really all about rearranging the molecule. So it's an isomerization of citrate. And so inside of this enzyme, we have a 4-iron and a 4-sulfur cluster inside of the enzyme in order to facilitate this reaction. And so what you'll notice is that you have kind of a twofold mechanism that's going on here. If you look at our very first one, right, what we need to do here in this first half of our reaction is we're going to remove a hydroxyl. So here we remove hydroxyl, right? But in our second phase of this step, we're going to do a hydration reaction. Right? And so what we end up with is this hydroxyl shifted down, right? We remove it and then we stick it back on, but just in a different place. So you remove it, you make a double bond. It's in the cis orientation, and so that's cis a-Ketoglutarate, right? And then when you add back that hydroxyl, you're just adding it back to a different carbon, okay? And so it's all about making sure that hydroxyl goes in the right place. Okay, now you have isocitrate. That's what you made, right? It was an isomerization. So you're just making, it's the same carbons, the same everything. All you've done is basically rearranged it. Now you're going to go through a dehydrogenase reaction. So this reaction, again, is one of those irreversible steps. This step is very, very important because this is the rate limiting step of the entire cycle, okay? This is highly, highly regulated, it's highly favorable, and there are a lot of different ways that this is regulated. So let's look at what happens here. So remember, these are the carbons here from the acetyl group, from acetyl-CoA, right? From the acetate group of acetyl-CoA. That's where those came from. So now we're kind of watching what's going to happen to this molecule. So as we go through this dehydrogenase reaction, what's going to happen is dehydrogenase, we're going to lose hydrogens, right? So from here to here, we're going to have an oxidation, which is a loss, in this case, of hydrogens. So if we look here, this is a hydrogen and this is a hydrogen that we're going to lose. Look at that carbon. We've lost 2 hydrogens from that carbon. Where did those 2 hydrogens go? They are right here. One's on NADH, and then one is on H+. And so this is where we're converting NAD+ to NADH, right? Here's our electron carrier. Now what's going to happen is that we're going to undergo a decarboxylation reaction. So from here to here is a decarboxylation. What does that mean? That means we're going to lose carbons. So here's our CO2 that we're going to let go of. So we're going to lose one of our carbons, and this is the carbon that we're going to lose. Because when you see when it goes here, that same carbon has lost a carbon dioxide. So you'll notice it is not one of the carbons that was involved in the acetyl-CoA molecule, right? And so we make, in the end, alpha-ketoglutarate. This oxalosuccinate is very, very intermediate, very transient, does not hang around for a long period of time. But alpha-ketoglutarate, sometimes called 2-oxaloglutamate, TAGLUT glutarate. So just because sometimes you'll see it in two different names, but it's the same thing. So this is kind of cool because this right here is a shared metabolite between the citrate cycle and some other pathways, right? So it's a substrate and it's a product, right? It participates in amino acid metabolism. So we're going to see a lot of crosstalk between the citrate cycle and some of the other pathways, which is really pretty important. This pathway is highly regulated, like we said, and it's highly regulated by, let's see, where can I put this? Regulated by ADP, right? And calcium. Okay, so these are positive effectors. If you have lots of ADP in the cell, then that means you need to start to generate some more energy for the cell. And so this will promote isocitrate dehydrogenase. Whereas on the other hand, our inhibitors, inhibitors are ATP, right? That makes sense. And then NADPH, right? These are negative effectors. And so it's really to make sure that if you have lots of energy in the cell, you could be using that substrate for something else. And we'll figure out what that something else is in a little bit. Okay, reaction 4. This is our second oxidative decarboxylation reaction, right? So this is another one of those. Our reaction 4 is highly favorable and it is irreversible, right? Another one of those large negative delta G values. And so this is the same exact catalytic mechanism as pyruvate dehydrogenase. So all the details that we went into in pyruvate dehydrogenase, so this is like pyruvate dehydrogenase with the E1, E2, E3, and needing TPP, lipoamide, and your NAD, right? All that kind of stuff, that's all exactly the same that you need. And so this one, right, because it has all those same components, this one is also regulated by the energy charge of the cell. And remember, energy charge of the cell is really the ratio of ATP to ADP, right? And so this is regulated in exactly the same way, right? And so we go from alpha-ketoglutarate to Succinyl-CoA. Now our Succinyl-CoA, now that we have our CoA molecule attached on there, now we're going to be able to go through and actually produce an energy storage molecule, right? We're gonna take that and we're gonna go produce GDP. So our reaction 5 is Succinyl-CoA Synthetase, and THEASE. Sorry about that. And this is a substrate level phosphorylation, right? Because we're going to take a phosphate group that's floating around, right? And we're going to stick that onto GDP to make GTP. So we're going to go from Succinyl-CoA to succinate. Now, whenever we make GTP, what's actually going to happen in the cell is that this GTP is going to react with a DP, right? And this catalyzes a reaction to GDP plus ATP, right? Because this is really, ATP is really the energy currency of the cell, right? So we have our enzyme nucleotide, okay, right? Nucleotide. I don't know if I'm going to be able to, diphosphate, nucleotide diphosphate kinase, right? And so that's the enzyme that catalyzes that reaction. And it's really important that it does that, and it does that very, very, very quickly, right? And so you got to think about why this transfer of phosphate occurs, right? When we start over here, what we have is a thioester bond that's storing quite a bit of energy inside of that bond. It's like negative 34 about kilojoules per mole, right? And so the amount of energy that it takes to transfer a phosphate group is a positive 30 kilojoules per mole. But when we look at this entire overall reaction, right, what we're going to get is a delta G in standard conditions of negative 2.9, right? So it's not just a subtraction between the two, right? There's a mechanism that's required in order to do this. And your book refers to it as pass the hot potato. So you have to go through, you notice we don't draw these intermediates in here. We just kind of say, it's a really long line and some things happen. So there are several high energy intermediates in here. So in this area here, we have several high energy ER. Intermediates, right? And the reason we have a few high energy intermediates is because we want to ensure that the energy that's gained by cleaving that thioester bond is not lost before being used to generate GTP. Once we get it to GTP, it's pretty stable. We can convert it with nucleotide diphosphate diphosphate kinase in order to make that ATP. But we really need to get it to the GTP form. So we do a bunch of high energy intermediates, and I think your book shows you the intermediates and how they form inside the enzyme pocket, but you don't need to know that. But you just need to know that there are intermediates in order to harvest that energy, right? In order to not lose that energy. Okay, reaction 6. Reaction 6 directly links our citrate cycle to the electron transport chain. So links to the electron transport chain. And this is through a redox conjugate pair of FAD and FADH2. So we have this pair of high energy carrying molecules that are covalently linked to our enzyme Succinate Dehydrogenase. So they're covalently linked to this enzyme right here. And so this protein is actually associated with the inner mitochondrial matrix. And so what we're going to see is that once we transfer these electrons, when we go from succinate to fumarate, we're going to change FAD to FADH2. So we're going to pick up those electrons, and later those electrons are going to pass from FADH2 to the electron carrier coenzyme Q, which is in complex 2 of the extracellular of the electron transport chain. So you don't have to know about the electron transport chain yet, but I'm trying to bring it up now so that when we get to it, you're going to go, oh yeah, I remember where the citrate cycle made those FADH2s, and that's where it's being used. But you do know that this is the reaction that links directly to the electron transport chain. Okay, so now we're sitting at fumarate. We made another electron carrier, and so this 7th reaction is really a hydration reaction. And so if you look at fumarate, what we're going to turn fumarate into is malate. And so this is a really interesting reaction because you're making an L isomer of malate. So anytime you have a chiral carbon, so a carbon with 4 different things attached to it, it could be in either the D or the L orientation. And so if you look at the hydroxyl group, we see the hydroxyl group is on the left. And so we can see that this is the L isomer of malate. And this is this is like one of the only reactions where you're looking at an L isomer. Most of all the other reactions inside the cell are D isomers, but the body's actually designed to only work with the L isomer in the citrate cycle, which is kind of cool, right? But it's just a hydration reaction, right? And it's kind of a two-step where in the first step, we're going to add the hydroxyl, and in the second step, we're going to add the hydrogen ion in order to hydrate that carbon, right? Okay, so the next one is malate dehydrogenase. Malate dehydrogenase is another oxidation reaction. We're going from malate to, guess what? We are regenerating our initial substrate, which is oxaloacetate. So this one is another dehydrogenase, so that we are undergoing conversion and movement of those electrons, right? So we are going to lose some hydrogens in this reaction. So the hydrogens that we lose, highlight them, are here and here. And where do they go? One goes to NAD, so we make NADH, and then one becomes a hydrogen ion. And we end up with a carbon double bond to an oxygen. So this is oxidizing our hydroxyl group of malate. It's going to remove the hydrogens, right? So now that we kind of have looked at every single one of our reactions, what I want to do is I want to take a quick look back at our initial, if I could get back to, okay, our initial reaction. So one thing that we've done in the past is we want to know exactly how these different molecules are made and react within the cycle. And so what we've done is we've radio labeled certain carbons and said, okay, what's happening, right? What happens when our pyruvate actually turns into acetyl-CoA, and what happens to those carbons that were there in acetyl-CoA? Well, here are the carbons that were present in the acetyl-CoA, and what you're going to notice is that these carbons don't go anywhere, right? So when we go and we form citrate, we transfer those two carbons. We're going to undergo that isomerization reaction and make isocitrate, right? Now, in our first dehydrogenase reaction, we're going to lose a carbon. Here's a CO2 right there, but we didn't lose these two carbons. If you look down here, what we've done is we've lost a carbon that was on the initial oxaloacetate molecule. And every time we're losing carbons, we're losing two carbons, right, in this whole cycle. Well, how many carbons do we add to oxaloacetate in the beginning? Two, because we lose two as carbon dioxide in the rounds of the citrate cycle. And so it's a nice balance. I like really like the citrate cycle because it's really, I don't know, highly organized, and I don't know, I like that sort of thing. Okay, so go through the cycle and understand what reaction's happening and what's important to get to our final product, to get back to oxaloacetate. So hopefully we covered all of this in class, but if we didn't, you've got this video to go back to. Have a great day.